You're listening to Strange by Nature, your guide to the strange, weird, unbelievable, and improbable wonders of the natural world.
Hello, everyone, and thanks for being here today. I'm Kirk Mona, and joining me are Rachel Gimza and Victoria Thompson. We're all naturalists who together have scoured the world for weird and wonderful wonders, just to please your mammalian brain's desire for novelty. Isn't that nice? Let's do this.
Well, hello, everybody. Welcome to the show. Good to see you guys as always.
Good to be seen.
I am starting things off this week.
Or, heard at least.
Well. All right, so I'm starting things off, and I'm going to talk about a beautiful region of the world. If you spend any time on sandy beaches in tropical or subtropical areas of the Indian and Pacific Oceans, you may notice a strange phenomenon. Tiny spheres of sand that are scattered across the beaches.
Okay.
Little balls of sand, but not randomly. They form radiating lines and sunbursts, sometimes spirals or concentric circles. These works of art last for a few hours until they're washed away by the next tide. And these patterns are the work of small crabs.
Oh.
Love a crab.
Yeah, known as sand bubbler crabs.
That's so cute.
Yes. There are various species in the genuses Scopimera, I don't know, and Dotilla. The crabs are quite small. They're usually about 1 centimeter wide across their carapace.
Mm-hmm.
And otherwise they look, very crab-like. They just look like typical crabs. They are sand-colored and they camouflage really well against the beach. And they make small burrows where they hide during high tide. When the tide goes down, they emerge from their burrow and look for food. Well, what do sand bubblers eat? They eat sand.
Of course. Why wouldn't they eat sand? They're only sand bubblers.
Like small children, they eat sand.
Nightmare.
Of course, they are not actually eating the sand grains because that is hard rock. What they eat is microscopic organic material from the sand. Detritus, tiny animals, single-celled creatures. That's what they dig. Their front claws point downward and are specially adapted to scoop up sand into their mouths. They also have specially adapted mouth parts that are lined with bristly setae, sort of like hair-like fringes. They store water in their gills, and as they scoop sand into their mouth with their claws, they vibrate the water through the sand and use the setae to filter out the organic material from the sand, in between the sand grains.
Okay.
So then they swallow the food, but as they release waste sand, clean sand, it forms a small ball, and when it reaches a certain size, they spit it out on the beach and put it behind them.
Yeah, sure. Why not?
Very cool.
The sand balls can be 1 to 9 millimeters across, depending on the size of the crab. They just push it back, and then they move on to the next bit of beach. Okay.
Makes sense, I guess.
Yeah. What about the patterns? Well,
why are they in fun patterns, Victoria?
Yeah.
The center of the pattern is always the crab's burrow. And the crab moves forward as they filter the sand. They place the balls behind them. When they've gone as far as they like, they head back to the burrow and start again on a different trajectory. This creates the starburst pattern. And by making these balls, the crabs then have a clear visual representation of sand that has already been processed.
Nice.
So they're not going to waste time and energy on areas that have already had all the nutrients extracted.
Right.
Yeah.
Okay.
Brilliant.
The pattern is going to vary depending on the species of crab and also how the nutrients are distributed. They're not going to spend as much time in a location that's nutrient poor. They're going to spend more time in an area that has a lot of nutrients. And there are also numerous crabs on the beach usually, so their patterns run into each other and overlap. So it can create these kind of intricate combinations of patterns. Of course, the crabs have a limited amount of time to do their work. They can only feed at low tide, and as the tide comes back, it will wash everything completely smooth again, but also distribute more food in the sand for them to eat.
There you go.
However, this crab is adapted to breathe air. So
yeah, you said that the center of those starbursts is his burrow, and if the tide is washing in and going over that, then they're underneath the water, Victoria.
Indeed, you've put your finger on the problem, Rachel.
Woo-hoo.
So what they do is they head back into their burrow as the tide is coming in, and they use their little front pinchers to build a roof of sand over the hole so that it creates an air bubble.
What?
Yep. And then they hang out inside the air bubble after the water comes up. It just traps that little bubble under the sand.
Okay.
And they hang out there while the tide is high, breathing in the little air bubble until the tide goes back down.
They make themselves little bob. They're doing the canoe trick in so many movies that humans use.
Yeah.
The bubblehead
with
charm
sand.
or something like that.
I mean, they are called sand bubbler crabs.
You are correct.
I assumed the bubbler like referred to the
bubbles.
the little balls they make. Yeah,
I think also that, yes.
Yeah. Wild.
that is
But
awesome.
speaking of breathing, the other really weird and unique thing about these crabs is that they actually breathe through their legs.
Oh, okay. Hold on.
What?
Yeah.
Excuse?
Yes.
Come again.
The top segment, sort of, if you make an analogy to humans, the thigh of the legs has these round membranes that are kind of translucent. And for a long time, scientists didn't know what they were for. They thought maybe it had something to do with hearing and sound. But based on some experiments, we now know that these are used in gas exchange. They're called gas windows. And they can exchange gas with the air through these membranes on their legs.
That's wild.
It's just
So cool.
so strange and random.
Okay.
Why not? I mean, they're using
Sure.
their gills to flood water through the sand that they're eating.
Yeah.
So
Why not?
nature is amazingly inventive.
That's so cool. I'm just sitting here like they're breathing through their legs.
Yeah.
Okay.
Yeah.
And
Why
they're
not?
little artists without any conscious
Concept
intention
of
to make
art.
art. Yeah.
That's
I-
so cool.
Yeah, I randomly came across these, I think maybe on Reddit or something, and I was like, wow, I just need to talk about those. I got I got some information this week from A to Z Animals, which had a very helpful article. There was some good information from the Living Waters Museum of India and also Hong Kong Green Power, and also some background information from Wikipedia. We're going to take a little break now, and when we come back, it'll be Kirk.
Thanks, Victoria.
You're welcome.
Hey everyone, welcome back. Now, Victoria,
Yes.
a few weeks back, you shared a really just lovely, special Mother's Day story with us. Do we
yeah,
remember
so
that one?
heartwarming.
Yes. So
This
special.
was the story about mother spiders giving the ultimate sacrifice by having their babies devour them.
Yep.
Now, you claimed, Victoria, that you had no idea the mother eating story was actually going to come out the week of Mother's Day. Is that correct?
I had not thought that through. It's true.
That is
Excellent
outstanding.
job.
Well, this week, I was looking through some of my story ideas, and I came across one that I had saved quite a while back, and it just so happens to dovetail nicely with your story of matrophagy. And I thought about saving this for Father's Day, but with Victoria's story fresh in our minds, why wait, right?
And Father's Day is not that far away.
It's not that far off, right? So here we go. This week,
You're a dad,
I bring you a
it's
story that is quite the opposite of Victoria's, not just because it involves fathers, but because it involves fathers eating their children.
Oh, lovely.
Kirk.
Oh,
Kirk.
that's
Strap in.
even less
Here we
heartwarming
go.
somehow.
Now look, that sounded,
no.
I know, that sounds pretty graphic. It isn't quite as dramatic as I made it sound.
Right.
The fathers in question here aren't really eating their children. They're eating their potential children. We're talking today about fish. Specifically, the Beau Gregory damselfish, which is found in the Caribbean. And I will say, though, this behavior that I'm going to be talking about isn't limited to just them. They're just the ones that happen to be in the research article that I was checking out. This is something that I guess happens a lot in fish, and it's quite interesting. When the mama damselfish lays her eggs, she then swims off to do other fish things, and she leaves dad in charge. And dad will frequently eat some of the eggs he has been put in charge of guarding.
Huh.
Well, that seems like you shouldn't leave dad in charge.
Yeah, right? I mean, guard duty turns dad into a cannibal that eats his own kids is not really what you think would be the optimal solution here. And this seems incredibly counterproductive since he's eating the very thing he was meant to protect. It actually kind of reminds me of Grogu eating the eggs that they're supposed to be protecting and delivering in that one episode of The Mandalorian, if you're a fan of that. Eating
Spoiler, I guess.
Not at this point. Eating the things you're meant to protect, pretty weird. So scientists have long known about behaviors like this, but haven't necessarily known why it occurs. And there have been a few ideas thrown out there, though, that seem to make sense. So one theory was that dad is eating because he needs calories. And it's actually better to eat a few eggs and stay on guard duty than to go forage for food and come back to find all of the eggs have been eaten by predators. Right?
Sure. I could
Right.
So
buy
it seems
that.
like a good trade-off there. In this scenario, mom is essentially laying extra eggs as food so dad can be well-nourished and fed without having to abandon guard duty. And that actually seems like pretty solid theory.
I mean, it's
not bad.
It's not that different from when mothers of different species lay extra unfertilized eggs for their other babies to eat, which is something that happens
right.
pretty frequently.
So there's precedence for something like this. So when you hear an explanation like that, you go, yeah, that could be. Well, researchers tested this theory by giving male damselfish in Jamaica extra food. The fish that got extra food did grow larger, but the mortality of eggs did not change. And so it seems like they aren't eating the eggs because they're hungry, which is pretty interesting. So then the researchers concluded that there must be something else going on if they're not doing it out of hunger, because they didn't kind of need the calories and they still ate the eggs. So researchers suspected it may actually have to do with oxygen.
Oh. That's not what I was going to guess. I was going to guess that the eggs were like defective or infected or something.
Interesting
I was
ideas.
going to
Rachel.
ask if the eggs, if they knew for sure that all of those eggs were fertilized by him, if they were competitor. Males.
I think the assumption was that they're all his eggs. You know. So, like I said, the actual solution is probably actually oxygen, which seems wild. Here's how they tested this theory out. So first they put up clutches of eggs into tanks where they could control the oxygen levels in the water. They kept the oxygen the same in each tank, but they varied how many eggs were in each tank. In just 48 hours, the tanks with fewer eggs showed faster development. So essentially, there was more oxygen per egg when some had been
Oh,
removed.
so oxygen is a limiting factor.
That's interesting and all, but
Okay.
is that really why Dad is nibbling on their own young? Well, it's time for experiment number 2. So this time they added a male fish to each tank, but they kept the same number of eggs in each tank. And the variable this time, again, was how much oxygen was in there. And
Okay.
what was really interesting was to see that the lower the oxygen level was, the more eggs the male fish would eat, which thus increased the surviving egg's chance of survival. So putting those two together, we know that lower oxygen levels makes eggs that are healthier and develop quicker. And we also know that when the oxygen levels drop, the dads eat more eggs. So that's just really, really fascinating.
Lower oxygen
Yeah.
levels make them develop?
Lower oxygen levels make the dads eat more eggs so that
So-
there's more oxygen per egg that's left.
Yeah, okay.
Got
You
it.
said, I think you
Yeah.
misspoke earlier, or maybe I misheard. Anyway, go on.
All right.
Good
so
clarification.
Yeah. So this would seem to indicate that cannibalism isn't driven by hunger in this case, but rather by a pressure to reduce the number of offspring in low oxygen water to increase success of the offspring. And
Huh.
now this is wild if you just come at this from like, why did they just all of a sudden decide to test oxygen levels? Like there's
Right.
so many other things that it could be. Then you guys
Yeah.
named a couple.
Temperature.
It seemed that they just kind of pulled this out of thin air. Like if I found out that the cannibalism wasn't due to hunger or eating while staying on guard.
I'm sorry.
My first,
Hold on. Thin air is
I know.
oxygen.
got to get past
Sorry,
it.
keep
It's
going.
all right. I wasn't going to acknowledge it. It was not intentional. Like I was saying, if I had learned that it wasn't hunger or eating while staying on guard, my first thought would not have been, oh, let's test for the oxygen levels in the water.
Right?
So
No.
where is this coming from? And where it is coming from is that many species of fish, while watching their eggs, actively use their fins to fan the eggs and increase the amount of oxygen they can receive from the water. So this is like a known thing that fish do.
I
And
guess I have seen crayfish do that with their eggs.
okay, cool.
Yeah.
Well, these fish, these damselfish, do not fan their eggs. So the speculation was that perhaps they had found another way to increase oxygen, and it does appear that that is the correct solution. And look, I've said this on the show many times, but it is something I say a lot while teaching. One of the things that I find so fascinating and interesting about nature is that evolution has found multiple solutions to the same problem. And
Right.
one of them isn't right and the other wrong. They're all just solutions that are being tried out currently. Long term, some are going to work out and some don't. And sometimes there are multiple solutions that are equally effective and all of them persist for millennia. And it's just so cool that we can look out at the world right now and see this spectacular and fascinating variation for ourselves. And so this is just another example of that. And I think it's fascinating.
That's so cool.
Yeah.
I
It
love
is
that.
it is fascinating.
So my sources were kind of twofold on this. There was the journal article, Filial Cannibalism Involves Survival and Development of Beau Gregory Damselfish Embryos. That was in the Proceedings of the Royal Society B Biological Sciences. And also a story in Science News by Susan Milius called Eat the Kids. Are cannibal fish just refreshing the O2? Great title. Great title.
Beautiful job.
So yeah, cannibal fish actually doing a favor for the kids. We are
Wow.
going to take a short break here, and when we come back, we will have Rachel. This week's episode is brought to you by the Society of Strange. That's what we call our patrons over at patreon.com/strangebynature. Society members directly support the show they love, and depending on their level of support, get fun perks such as bonus content on select shows, insider photos, water bottle stickers, and here's a big one, every level of support gets every episode 100% ad-free. Come join the other lovers of the show, help keep this strange show going, and get those ad-free episodes. We'll see you all over at patreon.com/strangebynature.
Welcome back to the show, everyone. So I love that we're just in a wild group this year. My goodness, we're always in a wild time. So speaking of, when you are a scientist and you're doing any sort of experiment,
Okay.
what do you do when something goes not as you expect?
Oh, you set up an experiment where you check for the oxygen level in the water.
Right, obviously. Duh.
No.
You
when it
try it
doesn't go as
again.
expected. That's a great answer. Yeah, you could
So
try
you try it again and you get the same result.
You reevaluate your hypothesis.
Ooh,
Yeah.
fascinating. All right,
Could
so
be an option.
enter Dr. Richard Jorgensen. We're going back to 1984-ish, okay? During the 1980s, all right? This is the beginnings of the beauty that is DNA and learning about RNA and genes.
Yeah.
We are
Good times.
going over to a DNA Plant Technology Corporation. Is what it was called at the time. We're going out to California. And what is happening is they're trying to learn a little bit more about just gene sequencing. This is still really new at this point in time, okay? So
Okay.
no one knew everything, but they were able to identify certain genes. So Dr. Jorgensen and his team, they were going about trying to figure out, the idea was that they were trying to figure out herbicide resistant crops. So
All
things
right.
that were more desirable and wanted for, agriculture, which is super important. They also wanted to be able to create new varieties for gardening and making things prettier and wanting to make things the way we wanted them, So
Okay.
this is the wild west of gene sequencing and gene manipulation, okay?
Early days, yeah.
Early days.
Mm-hmm.
Now, here's the thing, is they were going about this and they were trying to create these experiments. So they decided to use petunias, all right?
Okay.
All right.
Why not?
All right. Right. Well, they have a wide variety of colors, and they were trying to figure out, okay, if
Classic
I do this,
experimental
how can
plant.
I make more this purple petunia? How can I make it even more purple?
Sure.
So they
Sure.
took this one particular species of purple petunia, and they're like, okay, well, we know what gene is causing that creates the color purple in this petunia. So their
Okay, yeah.
idea is, okay, if we introduce something that stops it from, they want to oversaturate that gene, so that way it makes it as purple as possible. Okay, so what they
Like it'll
did
overproduce more of the purple pigment or
exactly,
something.
and just make it the most
Okay.
purple petunia that could possibly ever exist. Okay.
Seems like reasonable, like that might work.
Right? Because you know the gene, you know what you can do. And at the time, what you would use, when you were trying to deepen coloration especially, is you would use a type of sort of a virus, but not a virus, okay? So you were using you were using Agrobacterium tumefaciens, okay? So you were able to take this, and it
Sorry,
was
what is
pretty
this?
much Agrobacterium tumefaciens. It's a little it's like this little virus that they are able to use to introduce like a silence bit.
It can like edit genes.
Yeah, it's
Right,
able to edit
okay.
genes, but it doesn't cause the plant to get sick or anything like that.
Yeah.
Okay.
Okay.
So Dr. Jorgensen, he is using this and he's trying to deepen that color purple because he wants the most purple petunia that could possibly purple.
Gotcha.
And they and they introduce it because it's able to just increase the copies of this gene that causes the coloration. Okay. They were able to introduce more copies of this gene that allowed it to create more purple, and they did it in 3 different petunia varieties.
Oh, I can
and-
see where this is going.
Guess what happened?
It didn't work.
Not what they expected, I'm guessing.
Yeah.
They turned the rest,
What
the
color were they?
variations, white.
Oh.
White. Interesting.
No color.
Okay.
No color.
Whoa.
N-
Why?
Which they were like, what? Excuse? What do you mean?
It's not the purple is purple to ever purple.
No, and it should be. Like everything that they knew about genes, about the sequencing and everything was like cutting edge. There was no way for them to not be the most purple petunias to ever purple.
Is there any chance that the purple was just so purple that it was
It turned
stolen
white.
by the peculiar purple pie man of Porcupine Peak?
Maybe.
That is a
Perhaps.
very 1980s reference that
Yes, it was.
only certain people
I
who are fans of Rainbow Bright will understand. Or no, sorry, it was strawberry
I knew
shortcake.
that one.
Strawberry.
Yeah.
A strawberry shortcake, yeah.
I knew that one.
Okay.
Congrats. It's not as old as you think, Kirk.
It really is.
Anyway, so these they were able to like go forward with these plants and they were stable. They
Okay.
all had the same pattern. None like
They were
reverted
white.
back to being purple or anything like that. They all became and stayed white. They all stayed the same pattern.
So weird.
And they back crossed and everything, and it was weird. They were like, what is going on?
What happened to the
And-
purple?
What happened to the purple? The
Where'd it
only
go?
time the purple showed up is when that gene that codes for the purple was active. Because of this experiment, it opened up RNA. At this point in time, they really were not like aware of a form of RNA known as RNAi, which is a type of RNA that helps with. It's a type of messenger RNA that
Yeah.
helps
Okay.
like code for particular it helps pretty much make genes go from one spot to another. So if there's too many of a one particular gene, it stops it. I'm
Oh.
not explaining it extremely well. It is
So-
so nuanced.
If I'm understanding what you're saying, like this messenger RNA or whatnot is seeing that there's basically too many copies and going, oh no, something's wrong. And so it just stopped,
It pretty much
all
like
of them.
reduced and just got rid of everything. Yeah.
including the original copy.
Uh-huh. So they literally
Interesting.
Huh.
all went, Jorgensen and the team were like, what? But they kept going, and it happened to another group at the University of Amsterdam, and they were just like, what is going on? And they were able to find out that there were some subtle differences in some of the growing conditions, and they ended up figuring out that like the co-suppression of the gene was because of like it caused it pretty much what ended up happening, it caused like this gene to not be expressed in the way that it needed to, because the RNAi, which they just discovered was a thing, ended up like it's way too complicated to really get into. I'm gonna be real honest. Like, there was a lot of science that I don't think we have time for. But pretty much what ended up happening is they blocked the coding of the gene that coded for the purple in the petunias in the first place by accident. And before this,
And that's how we discovered RNA.
they had no idea about this form of RNAi.
That
because
form of R. So, like,
Yeah, we
the
knew
They knew about
non-purple
RNA.
petunia,
RNA.
Yeah.
Yeah, but they had no idea that RNAi is a defense mechanism, specifically for
Gotcha.
viruses. Because remember, the thing that they were using was a virus, technically, to help encode for more purple.
Right.
So
Right.
the RNAi was actually allowing and helping with the immune system of this plant. And they had no idea. And I will tell you, I barely scratched the surface for this. And y'all, it took them 10 years to figure this out.
So, like, you're trying to figure out that puzzle, and you know something's going on, but you're not sure what. I mean, that's like the best kind of science is like you get an answer, and maybe it's not what you expected, maybe it was, but you get more questions. And that's when exciting discoveries happen.
Exactly.
Absolutely.
It was absolutely wild, and it allowed more like research to be done when it came to gene modification as well, and for just gene editing, and for just what on earth is happening. We had no idea. It was just the beginning Wild West, and now we know a lot more just because a doctor went in to try to make the most purple petunia to ever purple. And I learned about this by accident this week, and I was like, I'm sorry, what do you mean the petunia turned white? That feels wild. Yeah, it truly is quite fascinating. It's a great read, and it's you can read it for free. I'm gonna say that right now. And it's really cool. So my sources this week, there was an article in 2019 called Patterns of Co-Suppression in Plants by Chris Surge in Nature. So Nature had had a great information. I got a lot of research from that. The original paper, the original paper, after they figured out what was going on, was the introduction of a chimeric calcone synthase gene into a petunia results in reversible co-suppression of homologous genes in trans, by Richard Jorgensen, Carolyn Napoli, and Christine Lemois. And that is also open to people to read. It is open access, which is great. It is the American Society of Plant Physiologists. And then Charged Magazine had some really great stuff. Same with UC Davis, which is where they are from, and doing some. He was a professor emeritus for a while. So with all of that, that is what I have for you all this week. The most purple plant that never was.
It's really cool that, like, you know, they came up with this way, like, oh, let's use a virus to, you know, a virus is because they can change the DNA. Let's use that. And it's like, yeah, we've had viruses on Earth, you know, for a long time. It's not really surprising that there are systems within our DNA to deal with that problem and go, no. I can tell something changed here. We're going to fix that. We're going to, you know, change that back. That's that is fascinating.
Yes.
It's very wild. And the fact that, like, they found all of this out because they were, like, testing plants and testing petunias, like, that really is what got me. But, like, this really was not a breaking point, but this really broke open this type of research in the scientific community when it came to genetics, which is really cool.
Very cool.
Yeah. I believe that brings us to the end of the episode, everybody.
Indeed it does.
Oh,
Indeed it
does.
does.
My goodness.
Oh, sadness.
Well, it is really sad. But we will be back again next week. So I will see you all. Well, I will talk to you all next week.
There it is. There it
We'll
is.
be in your
Bye-bye.
ears next
Bye.
week. Bye-bye.
Thanks everyone for listening to today's show. Be sure to subscribe. New episodes drop every Wednesday, and we love sharing this strange world with all our listeners. If you would be so kind as to leave us a 5-star rating and review wherever you get your podcasts, that would be great. It lets other lovers of the strange discover the show. You can reach us by searching for Strange by Nature Podcast on Instagram, Facebook, and Blue Sky. You can send us an email as well. Our address is contact@strangebynaturepodcast.com. If you want more information about the show, you can check out our website, strangebynaturepodcast.com, and you can support the show and get ad-free episodes at patreon.com/strangebynature. Until next week, get outside, stay curious, and embrace the strange.